I will start in a couple of minutes. Okay, so I'll share my slides. So welcome to the ninth live session of this course Structural Biology. Uh, in this session, I'll be talking about the summary of the things discussed in the videos of week 9, which basically covers visualization of macromolecules, uh, different techniques which were used in the old ages and modern techniques used now. So let's start. So why do we need to visualize biological macromolecules? So learning in science is abstract, especially when we are dealing with something we cannot see. Modeling by computers has provided us a way to see the things that were previously unseen and has transformed scientific research completely. So now we can visualize all kind of macromolecules like proteins and DNA using different softwares I will discuss towards the later end of this class. But before this, computer visualization softwares were developed. The molecular structures were presented by physical models using metal wires, rods, and spheres. For the first X-ray crystallographic structure of a macromolecule, myoglobin, Kendrew and co-workers built brass models. So this is a model built by Kendrew and co-workers. The scale was 5 centimeter per angstrom. So for each angstrom on the protein, the model had 5 centimeter length. And they were built and supported using these vertical rods. So you can see the protein, it has been imagined as a tube which was made of brass, and the scale was maintained. So for 1 angstrom on the protein, the model was 5 centimeter. And this entire globular structure of myoglobin was supported using these vertical rods. So this was a very primitive way of visualizing macromolecules before these modern softwares were developed. Now, with the development of computer hardware and software technology and different graphics programs, visualization and manipulation of these 3D structures have come very far. So these computer graphics help us to analyze and compare protein structure and to understand the functions of proteins. You know that the recent Nobel Prize, it was won by the software AlphaFold. So what AlphaFold does is that if you include a amino acid sequence, it can predict the structure of that sequence, three-dimensional structure. So the three-dimensional structure was determined by AlphaFold, that is shown in blue here. And the green is the structure determined from experiment like X-ray crystallography or NMR. So the RMSD between the two structures is very low. You can see the two structures are almost overlapping with each other completely with a very low RMSD of 0.8 angstrom. So AlphaFold's prediction accuracy is very high. This is again showing different regions where you can see the blue structure predicted by AlphaFold and the green structure shown by experiment are very close to each other. So AlphaFold not only predicts the three-dimensional structure of a protein based on the amino acid sequence, it also helps to visualize the protein 3D structure. You can see this is the structure of a protein with multiple helices. Here you can see a ligand which is binding to the protein. So you can see which side chains are interacting with a ligand. Like this is a histidine side chain, this is a phenylalanine side chain, and so on. So all this can be visualized using AlphaFold. And such visualization helps scientists and biomedical experts to study, understand, and engineer biological macromolecules. So only when we can see the molecule from close, we can modify the molecule to improve its binding or different functions. So that is another use of visualizing molecules. So we will go from 1968, which was the earliest when a visualization device was built by the biochemist Fred Richards. It was called Richards box or Fred's Folly. And it was the first visualization device, also known as an optical comparator. Here what happened, there was a solution of a crystal, a protein crystal, and electron densities resulting from the solution was calculated by the computer. And electron density maps were kept one after the other like this. So imagine you have a 3D structure and you have different slices of the structure. So the structure is sliced, there are different cross sections, and the electron density of each cross section is placed like this one after the other. Now what was done is a half silvered mirror was arranged like this. So this is a mirror which is superimposing the electron density map, this map, and a brass model. So a model of protein was created from brass and the electron density map was superimposed with the model. So what was done is that initially the model was determined, you can say, from initial extra NMR or X-ray crystallographic results. An initial model was built. Now, according to these electron density map, the model was refined accordingly. So, using this mirror, the model and the electron density map was superimposed and the model was refined to match with the electron density map. So somewhere if there was high electron density, a group having high electron density, like say the OH group, was added. And like that, the model was refined till it matched with the electron density map. And so now the protein could be visualized in the model. And as larger and larger protein molecules were solved, initially the scale was 2.5 centimeter per angstrom. And as larger molecules were solved, the scale became 1 centimeter per angstrom. So using this setup, molecules could be visualized in 3D using a model, and the validity of the model, like the model was refined using electron density maps, which had been obtained by X-ray crystallography. A second one, which Ken Drew had done, which I just showed in the previous slide, is, so this is a improvement of what Ken Drew had initially done, is just taken brass tubes like this to demonstrate the three-dimensional structure of the protein. An improvement of this was the ball and spoke models, where each atom was defined by a ball. You can see this red ball is an oxygen. Then there is a stick containing it, connecting it to a black ball, which is carbon. So this is a COO group. Similarly, white balls were used for hydrogen, blue for nitrogen. So each atom was defined by a ball, and sticks, also called spokes, defined the bonds connecting the atoms. So this kind of a iron or plastic model was built so initially Ken Drew had just built, you can see this white tube-like thing. That was the three-dimensional structure that Ken Drew had built. To specify the structure more, the ball and spoke model was superimposed on top of the structure. So now you can see the atomic resolution as well. And like in Ken Drew's case, the model was supported by these rods. Here also, you can see the entire structure is supported by vertical rods. The third physical visualizer was called Bidon's bender. Bidon Rubin invented a machine for bending wire to follow the backbone trace of a protein. So there was this machine, and steel wire basically was taken, and the machine would bend the wire to mimic the three-dimensional shape of the protein. So if the protein was alpha helical, you can see here the wire has been bent to mimic a alpha helix. Somewhere if there was a turn, you can see this is a one beta strand, beta turn, and another beta strand. So the wire was bent by the Bidon's bender to mimic the three-dimensional shape of the protein. These small backbone wire models from Bidon's bender were the most manipulable and portable models available at the time. Manipulable because it was easy to bend and manipulate the model, and portable because it was much lighter compared to the huge brass models by Ken Drew. The fourth kind of physical structure is the molecular sculpture. So there was a sculptor called Bathsheba Grossman who rendered proteins within glass blocks. So he would take a block of glass and make laser crystal. So basically using a laser, he would induce tiny fractures inside the glass block, and he would control the laser in such a way that the fractures inside the glass block mimicked the three-dimensional structure of the protein. So it is kind of, so you can see this wire model, it can, like while carrying it, this can be bent or destroyed. But when it is inside a glass block, you have kind of memorialized the model, and it will, unless the block itself breaks, the model is not affected. So this is one way in which the protein three-dimensional structure could be visualized. And many people have made sculptures in different ways out of wood or metal. You can see one such structure here. Now, as time went on, this kind of physical way of visualizing slowly improved. Till in the late 1990s, there was something called rapid prototyping. Efforts began independently by Michael Bailey and Tim Herman to use new engineering technologies of rapid prototyping to build physical models of proteins. So what happened is that there was a wax mold, and in that you can see this kind of a mold was shaped, where you can just pour the wax, and when you take it out, the structure of the protein is formed. So based on the structure of different proteins, different molds can be created. So you can see this is called mold printing. So suppose you have a protein which is mostly alpha helical, then the mold will be created in that way that when you pour wax in the mold and demold it to get the structure, you will get a complete three-dimensional picture of the protein. So this was called prototyping, because using this mold or prototype, you can get hundreds of structures. So earlier what used to happen is that you build one structure, and then like it takes too much effort to build more than one structure. But when this kind of mold or prototype began to be printed, you can get many structures in a short amount of time using the mold. And the mold, of course, can be reused. Now we come to the earliest computer representations of molecular structures, which began in the 1960s to 1970s. So as early as 1964, Cyrus Leventhal and his colleagues at MIT had developed a system that displayed on an oscilloscope the rotating wireframe representation of macromolecular structure. So this was the first computational representation of a structure. You can see the molecule backbone is shown by a wire frame. That is a thin wire-like structure, and it could be rotated. So displayed on oscilloscope means that the computer can be used to rotate the structure and look at it from different angles. So this was the earliest computer representation in the 1960s to 70s. After this, we come to the ORTEP structure. In 1965, Carol K. Johnson released ORTEP, which was a program to produce stereoscopic drawings of molecular and crystal structures. So basically what stereoscopic diagram is that you will have two identical looking images side by side, and when you look at the images through a special stereoscopic glass, the two images will kind of come together to give a 3D picture of the object with depth. So you are visualizing, so what is 2D on the page, once you look at it through the stereoscopic glasses, it will appear as 3D. You can imagine it as a 3D movie. You use special 3D glasses and the movie appears to be three-dimensional. So stereoscopic drawings is kind of like that. The full form of ORTEP was Oak Ridge Thermal Ellipsoid Plot Program. Oak Ridge was the place where it was first this program was made. Thermal ellipsoid. So basically around each atom you can see there is a elliptical structure. You leave the hydrogens. Each of the heavy atoms there is a elliptical structure. And so this is the program that will give you a structure like this. And this ORTEP rapidly became a favorite of small molecule and protein crystallographers to produce illustrations of structures for different conference presentations and publications. So you can see this kind of a structure. Different atoms were differentiated. So the program would tell you which is a carbon, which is oxygen. The program can also tell you what is the distance between atoms and so on. In 1966, this Carol Johnson, who had first released ORTEP, they produced stereoscopic drawings of myoglobin, vitamin B12 coenzyme, and polyalanine. So he showed the applicability of ORTEP in a protein, in an enzyme, and a polypeptide. One of the large advantages of the ORTEP was that the computer kept track of the atomic coordinates. So in the ORTEP structure, the XYZ coordinates of each of the atoms were kept track by the computer program, whereas they had to be manually measured from Kendrew model atom by atom. So this kind of a model, the XYZ coordinates of each atom had to be manually measured. So you can imagine how labor intensive it was. Computer representations took care of that, and the computer automatically kept track of all the XYZ coordinates of all the atoms. Next, we come to MolScript. MolScript is a program for creating schematic or detailed molecular graphics images from molecular 3D coordinates, usually but not exclusively protein structure. So the user supplies an input file, also called the script, which specifies the coordinates, XYZ coordinates, that is the 3D coordinates, and the user specifies that what kind of objects to render, meaning that they will specify that the helical part has to be shown separately, or the beta sheet part has to be shown, and the appearance of the object. So whether you want the helix in a ribbon, so this is called a ribbon representation, where the helix is appearing like a ribbon. There are other representations. You can specify what kind of representation you want. And using the XYZ coordinates, MolScript program can generate the three-dimensional structure of a protein. So these are representations that the MolScript program has generated based on the coordinate file. You can see this is a alpha beta barrel protein. So there are alpha helixes, there are beta sheets alternating, and this is an alpha beta barrel. This is a bundle of 4 alpha helices. This is several beta sheets, one after the other. This is called an alpha horseshoe. So a wide range of three-dimensional structures can be represented by MolScript. Next, another program is called Persistence of Vision, or POV ray visualization. It is a cross-platform ray tracing program that generates images from a scene, text-based scene description. So what this means is, imagine you have all these balls stacked one upon the other, and the program will trace the outline of these balls using a camera, and it will describe what it is seeing. So it will say that from one angle, the balls look like this. From another angle, they look like this, and so on. So the camera will trace the outline of the balls and give a three-dimensional view of the balls in different, from different angles. And using that that description that the camera has obtained, the computer will generate the images. So ray tracing occurs from the camera that will first look at the image and trace the three-dimensional outline of the image from different angles to the scene. That will be converted into computer images. And that is called the persistence of vision, or POV ray visualization. Now we come to, just one second, TAMS, which is Teaching Aids for Macromolecular Structure. So basically, if you have to publish an atlas of all the computer generated space filled models in color, it is very expensive. So somehow these models have to be accessible to the general student in a cost-effective way. So a breakthrough occurred when an inexpensive cardboard viewer, so this is a cardboard viewer, was found for stereo slides. So let us first discuss what stereo slides are. So here you have a three-dimensional image of two trees, one behind the other. You can visualize this from two angles, the blue angle, which will give you an image like this, and the red angle, which will give you an image like this. So what the eye does, is that it will so when you are wearing the stereo viewer, it will superimpose these two images and it will form the correct image in the brain. So the brain should see a three-dimensional image of the object, not a two-dimensional image. So imagine that two-dimensional images are formed in the two different lengths. These form a pair of stereo slides. When you enter the stereo slides in this cardboard viewer and you look at it through putting your eyes in these two holes, the stereo viewer will kind of superimpose these images, and what you will see is a proper three-dimensional representation, and the depth will also be included in the representation. So you will be able to see one tree behind the other. So what TAMS or the teaching assistant did is that this kind of viewer was introduced where stereo slides can be inserted and you can look through the eye holes and get an idea of the three-dimensional structure of the protein. So TAMS could generally, it can accommodate a pair of slides of 35 millimeter slides in 2 into 2 inch mounts. So so suppose there are two slides, one would contain this image, one would contain this image. And when you are looking at it, no longer it will appear two-dimensional like this, it will appear properly three-dimensional. And this viewer folded flat for the convenience in the cover pocket of the three-ring binder in which TAMS was distributed. So basically there was a binder in which different slides of different stereo slides of different proteins was distributed, and the viewer to observe the stereo slides, this cardboard viewer, it folded flat and came with the binder. So this became a very cheap way for students to visualize molecular structures in 3 dimensions. Because at that time, it was not common for every student to have a laptop or computer. So if a student wanted to see the three-dimensional structure of proteins, this was an easy and cost-effective way to do it. Now we will come to the different computer systems used for visualization. So in 1980 to 1990, Evans and Sutherland, they introduced some computers which costed around $250,000 in 1985. They could display the electron density map. So you can see the blue net here, blue grid here is the electron density map, and the amino acid sequence can be manually fitted into the map. So the computer, Evans and Sutherland's computer, would give this blue grid, and manually you can choose different amino acids to fit into the grid. So you can see in this region, it is a tyrosine side chain which is fitting. In this region, it is a phenylalanine side chain that is fitting. So you can manually fit amino acids to this electron density map. And in color display would show a wireframe rendering of the amino acid chain. So this is called a wireframe rendering because again, it is like a thin wire-like structure. And in the computer, you can rotate it and look at it from various angles. So this was what the Evans and Sutherland computers did. Next came David and Jane Richardson's Kinemages in 1992. So David and Jane Richardson pioneered computer graphics representation of a molecular structure with a series of programs. The images formed by the programs were called kinemages or kinetic images, and they are set up to illustrate a particular idea about a three-dimensional object rather than neutrally displaying that object. So here just the neutrally the 3D structure of the protein is displayed. What the kinemages did is that they can highlight a particular idea. So suppose you want to find out which are the amino acids which are in the disallowed region of the Ramachandran map, like the Ramachandran outlier. So the kinemages help to illustrate the Ramachandran outlier. You can see they are shown in green here. Or if you want to find out where steric clashes are occurring, that idea can also be illustrated. You can see it is shown in pink here, where there is steric clashes between different groups. Similarly, other ideas can be illustrated using the kinemages based on what the author wants to emphasize. So you can have a proline ring which is puckered in an unfavorable way. That can also be illustrated. You can have bad rotamers. By bad rotamers, it means that certain torsional angles are not very favorable, but it is um, adopting that angle. So that can also be highlighted and so on. So what the kinemages or the kinetic images did, they added a different dimension to the visualization. Earlier in the Evans and Sutherland computers, it was just plain visualization. But what the David and Jane Richardson kinemages, what they did is that not only the visualization, but you can illustrate different properties of the three-dimensional structure, like where there are steric clashes in the structure, or where residues are falling in the disallowed region of the Ramachandran map, and so on. Next came Roger Sales Rasmol in 1993. So basically, there was a need for a program to allow entirely self-directed, neutral, interactive exploration of any available structure. This need was filled by Rasmol. Rasmol provided a much richer set of renderings. So you can see the renderings here were all wireframe. So a thin wire-like structure showing the entire three-dimensional structure. But Rasmol included space-filling renderings. You can see here each of the atoms are represented by their van der Waals area. So they are called space-filling models. It could also show shadowing and reflective surfaces. So you can see here, look at this atom. There is shadow in some places indicating it is towards the bottom, and it is bright in the upper region indicating that it is towards the top. So it is a much more practical 3D representation. Like here, it was a simple wire, but now using light color and dark color to represent shadows, this is a much more practical and easy to understand three-dimensional model. And it solved the problem of depth perception for computer representation of solid objects. So basically, solid objects represented by this, you don't know what is in front, what is behind. There was no depth perception. But using this kind of space-filling representation where light and shadow was used to explain the depth of the structure, this was provided by Rasmol. Further improvement in the speed of the shadowing algorithm made inter-processor communication rate limiting on the parallel processor computer. This enabled Roger to implement it on single processor computers such as Unix and later Windows and Macintosh. So basically what happened, as this depth perception program was improved, using it on inter-processor computers was difficult, and it slowed down the speed of the program. So Roger Sale later modified it so that it can be run on a single processor computer, like each student uses nowadays. And it is a multi-platform program, so it can be used in different operating systems like Unix, Windows, and Mac. Rasmol is very widely used throughout the world. It is especially popular with students and in locations where resources do not allow the purchase of expensive computer molecular graphics software. So that is another advantage of Rasmol, is that you don't need any expensive graphics software. It can simply be used with the normal windows that you use. Rasmol also has a CPK color scheme that it uses. So for carbon, it maintains a gray color, for hydrogen white, oxygen red, nitrogen blue, and so on. So these are the four main atoms. You can remember these four. Others are not that important to remember. So now we will talk about the PDB file or the protein data bank file. Just give me 2 minutes. So any PDB file can be searched by typing the 4-letter PDB code inside the search box. So you have protein data bank or PDB, and in that you have a search bar you can see here. If you type the 4-letter PDB code, So PDB codes generally have 4 characters. The first one always has to be a number, and the other 3 can be alphabet, or they can be numbers. You type this code inside this search bar, and you can download the 3-dimensional structure of that protein. It also has detailed information about the structure with that PDB. So you can see here 3HP1 has been inserted in the search bar, and it is the PDB code for the human DCK. So it is cytokine kinase, and it is in complex with some nucleotides. So detailed information comes. It will tell you about in which paper this protein has been reported. It will mention that journal of the paper, the DOI, and it will also give you the abstract of the paper. So it will tell you can access this paper to find out more about this 3-dimensional structure. It will also tell you about the C2, that this has C2 symmetry, the stoichiometry, and so on. So different data about the structure can be accessed in the protein data bank. To display different features related to the PDB file, you can click on display files, and you will get different features, like you will get the FASTA sequence, which is the amino acid sequence. I will discuss that in the next slide. And you can download the PDB or protein data bank format of the file. I will show that to you after some time. So I was talking about the PDB file, and you can basically go to the protein data bank. I'll show you. So let us think. We want to find the PDB. So you can have a question that suppose we don't know this 4-letter code, then how can we access the structure of that protein? So basically then what is done? So imagine I want the structure of myoglobin. I can just write myoglobin PDB. And you can see the first option itself, if you click on it, it opens up the protein data bank where you can see the 4-letter code of myoglobin, and you can see it will tell you about the paper in which myoglobin was reported. And it will also tell you this structure, it was determined from which method? From X-ray crystallographic diffraction. And the resolution was 2 angstrom. It will tell you the authors who deposited it and from which organism it was isolated, and so on. So you can see the 3-dimensional structure of the protein myoglobin here. Now, that is how you can access, even if you don't know the 4-letter code, you can access any protein. So when you go to display files in the, I'll show you again. When you go to the display files option in the PDB, you have something called FASTA sequence. So what is this FASTA sequence? It is basically the sequence of, so in the field of computational biology, the FASTA format is a text-based format for representing either nucleotide sequence or amino acid sequence, in which the nucleotide or amino acid is represented using single letter codes. So if it is a nucleotide, ATGC will be used. If it is an amino acid, the 1-letter code of the amino acid will be used. So this is the FASTA format of this protein deoxycytidine kinase, and you can see the entire amino acid sequence of the protein is specified. So some characteristics of the FASTA file is that each line does not exceed 80 characters. And in the beginning, this symbol is used. You can see this symbol. And at the end, an asterisk or star is used. So these are some general features of the FASTA file. So the FASTA file is very simple. Initially, you have this symbol followed by a comment line which says that what is this protein, what is the PDB code, from where it has been isolated. This is followed by the single letter code amino acid sequence of the entire protein sequence. And each line has 80 characters. And at the end of this, there is a star. So this very simple format makes it easy to manipulate and parse sequences using text processing tools and scripting languages like Python, Ruby, or Perl. We don't need to go into that. Just you can remember that since FASTA is a very simple format with just text, so this is just a text format, it is easy to change. Suppose I change one single amino acid in this text format, I can just delete that letter and add another letter. And so it is very easy to change and manipulate the FASTA file. We will talk about some other file types now. One is the crystallographic information file, or CIF. This is a standard text file format for representing crystallographic information, and this was introduced by the International Union of Crystallography, or IUCR. So this file, what it contains, you can see the details in the IUCR website, but generally what it contains is that it will tell you what is the Bravais lattice. So this is triclinic, and it will tell you what is the space group. You can see it is P1, that is primitive, and the cell length. So the three sides of the cell A, B, and C, and the three angles of the cell alpha, beta, gamma, and also the volume. So all this is measured by, all this is mentioned by in the crystallographic information file. So it will tell you details of the crystal structure. And several programs can be used for visualizing the crystal. One of these include JMOL. The next kind of file is called a macromolecular crystallographic information file, or MMCIF. This is intended as an alternative to the protein data bank or PDB format. And in protein data bank, this is also available as a format in which you can download the structure. This is a flexible and extensible tag value format for representing macromolecular structural data. So you can imagine MMCIF as a combination of the CIF file and the PDB file. So MMCIF will basically have all this information about the crystal, and it will also have the XYZ coordinates of each atom. The fourth kind of file is called an MTZ file. So basically in an MTZ file, the electron density map can be read. So opening the MTZ file is slower because the electron density map needs to be calculated. But you can always recalculate the map using different grids to resolve the image further. So when you are reading an MTZ file, you have to specify the structure factor and the phase. So if you remember when we were doing the X-ray crystallography classes, for any crystal structure, there is a structure factor and there is a phase. So these have to be input to calculate this electron density map. You can see this blue grid here is the electron density map. And the default grid size taken for this is 0.75 angstrom for model building. But if you want a higher resolution, you can specify a smaller grid size. So that will give you a better picture, because smaller grid means higher resolution. But the program will take even longer to load the file because the calculation will be slower at higher resolution. Now, when the electron density map is loaded, you can manually choose which amino acid or which side chain or which residue fits along with the map. So there are some pre-specified models for which the electron density map is known. So you can check those models with the observed map and find out. So suppose you have the electron density map of alanine already specified. You can fit the alanine electron density map and see, so imagine it is matching with this region of your experimental electron density map, then you can say that an alanine residue will fit here, and so on. So if there is, you can go through a list of pre-specified electron density maps to see which region of the observed map fits with which structure, and like that the structure can be put according to the map. So MTZ file basically codes for this electron density map, which is the data obtained from X-ray crystallography. So you, in the protein data bank, there are, with each structure, right? So somehow I got disconnected, I guess. Right. So as I was saying, with each PDB structure, just one second. With each PDB structure, you have something called a validity report. So you can see here 3.3%. That means 3.3% of the amino acid residues are falling in the disallowed region of the Ramachandran plot. 15.2% of the side chain of the residues are in the disallowed torsion. So this is not a very good structure. So this is called a validity report. So in the protein data bank, with each protein structure, there is a validity report. Here you have two examples. One is A, which is a relatively good structure, and then you have B, which is a relatively poor structure. So you can see there are sliding scales, and it reports how a given structure ranks relative to all the structures in the PDB. So here you can see there are different methods for testing the validity, and there are different, you can see here, this is for protein, you can see in this structure, the amino acid sequence is given, and same for this structure. Now, there are different color codes. So when it is yellow, it stands, there is only one type of outlier. So one of the validity criteria is not being fulfilled. So that is yellow. Orange is when there are two types of outliers. Red is when there are three or more types of outliers. So if you just compare these two structures, you can see wherever there is yellow, if you compare these two structures, you can see in the left hand side, there is no red. So red will mean there are three or more kinds of outliers. So it is not a good structure because three or more of the criteria are being unvalidated. So in the left hand structure, you can see there is no red or orange. Whereas in the right hand side structure, so green is when there are no outliers. So in the left hand side, you have 44% green, and the right hand side 36% green. So green is good. It is when there are no outliers, and in that criteria, the left hand side is better because it is 44%. Then yellow is when there is one type of outlier. Left hand side, there is 11% yellow. Right hand side, there is 55% yellow. So the outlier in left hand side is much lesser, you can see, but the outlier in right hand side is much greater. It goes up to 55%. So that way also the left hand side structure is better. So this we are comparing chain B. If you are comparing chain A, you can see left-hand side 94% is green. So for 94% of the amino acids, there are no outliers. In the right-hand side, 75% of the amino acid have no outliers, which is less than 94. So this structure is better. So basically by using different colors, it will give you an idea of the validity. So green is the best where there are no outliers. Yellow is when there is one type of outlier. Orange, there are two. And red, there are three or more types of outliers. You will also see some amino acids marked by gray. Those amino acids are the ones where the atomic coordinates are not available. So, and for X-ray crystal structures, a red dot above the residue indicates a poor fit to electron density. So these red dots you can see, it is, I was just talking about fitting the electron density with the structure. So when the structure predicted is accurate, the fit of the structure with the electron density is very good. In that case, there will be no red dot. But when the structure predicted and the electron density are not matching, they are not fitting, then there will be a red dot. So clearly the right-hand side structure has so many red dots. So the structure predicted is not very accurate. So using the validation report, you can check how good a structure is in the PDB. So now we will talk about the basic format of a PDB file. In a PDB file, each line is 80 columns wide, and at the end of the line, there is a semicolon. The first 6 columns of every line contains a record name. By record name means it is specifying what that line is about. So here header is the record name. It is talking about the heading of the PDB file, which is generally the type of molecule. Like this is a transferase. So the type of protein molecule that is defined in the PDB file. The next record name is title. So it is the title of the PDB file. It is the crystals. So it is specifying what kind of transferase. It is the crystal structure of human DCK protein complexed with some other nucleotide. Then you have a record name, which is COMPND. So this record name is basically used to specify the name of the molecule, deoxycytidine kinase, the number of chains. So there are 3 chains and they are all named as A. So this is also specified. And in the top with the header, the date of release is written. That is when this crystal structure was deposited in the PDB, and the PDB ID is also written. So in a PDB file, if there is a spelling mistake in this column, then the PDB file will show an incorrect error when you are trying to open it in any visualization software. There are other information included in a PDB file, like the organism from which the protein has been derived and the common name of the organism. It will also give the journal in which the PDB was first deposited, and the resolution of the structure, which is 2.31 angstrom here. So you can see all this. I was just showing you. So when you open a PDB in the protein data bank, you can go to download files and you can see PDB format is written here. If you click on it, it will download the PDB file in PDB format. Let me just save it. Then I will open this. I'll first open it using Open With, and I'll choose Notepad. So you can see just what we read right now. So the PDB file of what we downloaded was myoglobin. So you can see the PDB file open here. In the heading, it is written the function of the protein, that is oxygen storage, And this was deposited on 5th April 1973, and the PDB code is 1MBN. Then you have the title, that is the stereochemistry of protein myoglobin. Then you have different information like the name of the protein myoglobin, the scientific organism, that is the sperm whale, common name and the scientific name. You also have the detail of the author and publication where it was deposited here. So you see, in these 4 lines where it was written JRNL, it stands for journal, you have the author, the name of the paper, page number, and so on. And then you have the different, there are different remarks about what kind of experiments and everything has been done. You can see what experiment was done to determine this structure. It was X-ray diffraction. As you go down the structure, there are many, I'll talk about the other details. So I was just talking about the PDB file, and I downloaded the PDB file for myoglobin by going to download files and PDB format, and I opened the PDB file in Notepad. So you can see what the PDB file consists of. It will tell you about the function of the protein, oxygen storage, the name of the protein, when the protein was deposited, and the 3-letter code. It will also give you it will tell you about the journal in which the protein was first mentioned. So this protein structure was deposited in a particular paper. It will tell you about the details of the paper, and it will tell you about what method was done to determine this structure. It was X-ray crystallography. You can see the experiment to determine the structure was X-ray diffraction. And it will tell you under what conditions the experiment was done. So solvent content and everything in the crystal. It will tell you about the symmetry, space group. So the different properties of the crystal. And as you go down, for each atom, you can see, you see this line. So it is saying that for atom number 1, you are talking about nitrogen atom, and it belongs to valine. And VAL is a 3-letter code for valine. And valine is in chain A. So suppose you have 3 chains in a PDB, 3 polypeptide chains. So valine is in chain A. Then it is telling you about the X, Y, Z coordinates of the atom. So I'll just go to that slide. So you can see atom, atom number. What is the atom? It is nitrogen. Then the 3-letter code of the amino acid to which the atom is belonging. Then A means chain A. 20 means the residue serial number. So in the polypeptide chain, it is residue number 20. Then you have the X, Y, Z coordinates. Then there is the occupancy. Occupancy means that suppose you have a phenylalanine and the side chain phenyl is occupying 2 different regions. So for some time it is staying in, so let us imagine a phenylalanine. Just ignore this. Let's do a valine. So imagine a valine side chain is staying in this orientation for some time, and then it is rotating and it is coming to this orientation. In this orientation, it is staying 70% of the time. And in one particular orientation, it is saying 30% of the time. Then XYZ. So this particular XYZ, it is staying occupancy 1 means 100% of the time it has only one XYZ orientation. Suppose it was staying 70% of the time in one particular XYZ, then this will be 0.7. And under it, you will have another different XYZ showing the other position, and that will be 0.3. So occupancy means how much time that particular atom is occupying that position. And thermal factor, you have already studied when we were doing X-ray crystallography. Thermal factor or B factor is an idea of the atomic mobility. High B factor means a very dynamic region. That is so you can see when you have here you can see when you have side chain, that is the red, B factor is high because side chains can move very freely. But when you have the main chain, that is green, you can see the green graph is much lower because main chain in amino acid generally does not move that much because it is part of the secondary structure. So main chain has low mobility. So B factor basically indicates atomic mobility, and that is what these B factor or thermal factor is showing. And again, you have the element symbol, which was given here. So this is how it should appear as, and I will go back to the PDB file I had opened in the text. You can see atom represents one atom. This is atom number 1. It is nitrogen. It is part of the amino acid valine. So VAL is written, belonging to chain A, and valine is residue number 1 in the peptide chain. Then you have the XYZ coordinates of valine. Then you have the occupancy. So 100% of the time valine is residing in this XYZ. And you have the thermal factor here, which is 0. So this valine is not moving at all. It is not a dynamic part of the protein. So that is what this thing is showing. Now we'll come back to the graph, and you can see, so this will represent all the atoms. Then you can have a record called CRIST1. This will give you an idea of the crystal structure. So we know in a crystal structure, you have unit cell, and the 3 sides of the unit cell form A, B, and C. And there are 3 angles, alpha, beta, gamma. So after the CRIST1 record, you will have 3 values, which are A, B, and C, the 3 sides of the unit cell. And you will have 3 angles, alpha, beta, and gamma. And finally, you will have the space group. So the unit cell parameters will be mentioned like this in the PDB file. So we talked about B factor, and then let us talk about TERD. So the list of atom records in each polymer chain must be terminated by a TERD record. So what this means is, if we go to the PDB file now, you can see all this is a huge list of atoms because it's a very large molecule. And at the end of the atom list, you have TERD, that is the terminal residue, which is number 1217, and it is glycine. That is what this is saying, that at the end of the atom list, there must be this record called TER. Now, whenever het atom is written, it refers to a heteroatom, meaning atoms which are not carbon or hydrogen. So nitrogen, oxygen, sulfur, these are all heteroatoms. And you have a record called connect, CONECT. What this connect defines is that, so if you have connect with 2 numbers after it, it is saying that atom number 1853 and atom number 1852 are connected by a bond. If you have connect with 4 numbers after it, that means these 4 atoms form a dihedral angle. So you have atom 1, atom 2, atom 3, and atom 4. So this angle is the dihedral angle. So if you have connect with 4 numbers after it, that means atom number 1856, 1851, 1854, and 1857, 4 atoms are connected by bonds like this, and there is a dihedral angle between them. If you have the word connect followed by 3 numbers, that means these 3 atoms form a angle. This is a normal 2-dimensional angle. So when you have the word connect followed by 3 numbers. That means atom number 1859, 1858 and 1860, 3 atoms form an angle between them. So this connect basically defines wherever there are bonded interactions, angles and dihedral angles. And finally, at the end of each file, there should be the word END or end. So if you go down, you can see the connect. This is defining that there is a bond between 1832 and 1830. This is defining there that 1238, 1235, 1239, there's an angle between them. And you can see at the last line of this PDB file, there is the word END or end. So this is what defines a PDB file. Just 1 second. Now we can, when we know the X, Y, Z coordinates from a PDB file, we can use it to calculate distance between 2 atoms or angle between 3 atoms or even dihedral angle between 4 atoms. So suppose you have 2 atoms, X1, Y1, Z1 and X2, Y2, Z2. You can use this formula to calculate the distance between the 2 atoms. Or if you have 3 atoms, X1, Y1, Z1, X2, Y2, Z2, X3, Y3, Z3. So 3 atoms shown by P1, P2 and P3. So first 2 vectors will be defined. One from P2 to P1. So one vector will be in this direction. You can subtract the coordinates of P2 and P1 to get the vector A. And another vector will be from P3 to P2. So it will be this vector. So that is defined by B, and you can get it by subtracting the coordinates of P3 and P2. So once you have these 2 vectors, we know that when there are 2 vectors, we can find out the angle between the 2 vectors by this formula, which is cos inverse. In the numerator, you have the dot product of the 2 vectors, and in the denominator, you have the magnitude of the 2 vectors multiplied. So we will just calculate that. So dot product means, so vector A is AX, AY, AZ. Vector B is BX, BY, BZ. Dot product simply means you multiply AX, BX plus AY, BY plus AZ, BZ. That is in the numerator. And we know to find the magnitude of any vector, we have to so suppose AX, AY, AZ, this is the vector coordinates. We want to find the magnitude. So we have to take AX square plus AY square plus AZ square and do the root to find the magnitude of A. Similarly for B. When we put it in this formula, we can find out the angle between 3 atoms if the X, Y, Z coordinates of the atoms are known. And similarly, if we have 4 atoms, we can find out the dihedral angle. So dihedral angle basic. So imagine there are 4 atoms, P1, P2, P3 and P4. Then the angle between this plane, which contains P1, P2, P3, and this plane, which contains P2, P3, P4, is the dihedral angle phi. And that can be found by this formula, where n is the normal vector. Again, we don't need to go into the derivation of this formula. Just remember that A vector is from P1 to P2, B vector is from P2 to P3, and C vector is from P3 to P4. So we will first take the cross product of A cross B and the cross product of B cross C. These new vectors, A cross B will be called NA, and B cross C will be called NB. And if we put NA and NB in this formula, we can find the dihedral angle between 4 atoms if the XYZ coordinates are known. So if the XYZ coordinates of the atom are known, which is the information we get from PDB files, we can find the distance between the atoms, we can find the angle between the atoms, and the dihedral angle also. Now there are different criteria to find whether a helix is being formed or a beta sheet is being formed. For the helix, the criteria is that from I to I plus 4 residue, if the distance is less than 3.5 angstrom, that means there is a hydrogen bond. You can see this dot is the hydrogen bond. So if the residue between the oxygen and the nitrogen, NH, at I and I plus 4 is less than 3.5 angstrom, then this hydrogen bond between I and I plus 4 will form, and that will lead to a helix. So we will follow the entire polypeptide chain. We will keep checking. So after I to I plus 4, we will check I plus 1 to I plus 5, then I plus 2 to I plus 6, and so on. And we will see wherever the hydrogen bond is forming. And when we see that the distance is becoming greater than 3.5 angstrom, at that point, there will be no hydrogen bond and the helix will terminate. So that is the criteria for alpha helix. Now for beta sheet. Beta sheet, there can be two different types of beta sheets. We have already discussed this, parallel and anti-parallel beta sheet. In parallel beta sheet, so this one, the residue I plus 2. So in strand, so let's call this strand 1 and this strand 2. So residue I plus 2 in strand 1 is binding with residue J plus 2 in strand 2. So let us call this residue as I. It is binding with residue J. So this will be I plus 1. This will be I plus 2. This is I plus 2. It is binding with J. So this is J plus 1 and this is J plus 2. Similarly, so you can see if we consider the nitrogens in the top chain and the carbonyls in the bottom chain, if I and J are binding and I plus 2, J plus 2 is binding, then it is a parallel beta sheet. Both are in the same direction. And here let us consider this as I and this as J. This is anti-parallel beta sheet where I plus 2, so this is I plus 2. This is I minus 2. Because the sheet is going in this direction. So if you go in the opposite direction, it will be minus. And this is J plus 2. So if I and J have hydrogen bonding and I minus 2, J plus 2 has hydrogen bonding, then they are in opposite directions and it is an anti-parallel beta sheet. So this is a criteria for beta sheet. Now there is a software called COOT, which stands for Crystallographic Object-Oriented Toolkit, or COOT. It is a macromolecular model building, model completion and validation software, and it is useful for modeling the protein using experimental structure. So here you can see from X-ray crystallography, you have determined the electron density map shown in blue here. COOT will display this electron density map, and you can fit an atomic model to the electron density map. So you can see a phenylalanine in this particular orientation shown by white is fitting into the electron density map. So like this, COOT will display the electron density map and the atomic model, and it will allow you to move the model in different ways like rotation, translation, and fitting to fit it to the observed electron density map. You can also check with the Ramachandran plot, and suppose you have an orientation where the Ramachandran plot, it is lying in the disallowed region. Then you have to modify the orientation such that it falls in the allowed region of the Ramachandran map and also fulfills the electron density map. So COOT can help you do that. And it is an easy to learn software with very user-friendly elements. And it is also freely available, so you can freely download it. And so that is another advantage of this software. Using COOT, you can read files containing 3D atom coordinates. So I just showed the PDB file has three-dimensional atom coordinates, XYZ coordinates of each atom. So you can open a PDB file in COOT. And then I'll just show you that once. So I have, one second, let me just So this is the, if you open Qt, a window like this will come. You can go to File, Open Coordinates, and open a PDB file. Let me just open a PDB file right now. This is a PDB file. I'm opening it. You can see Qt is displaying the file in a three-dimensional way, and using your mouse, you can rotate the structure to see it from different angles. You can expand the structure and look at it from different angles. So this is helping you visualize the three-dimensional. You can see this kind of a structure here. The atomic model is represented by default using a stick model we just saw, with vectors representing chemical bonds, and the two halves of each bond are colored according to the element at each end. So let us look at that again. You can see, let us say this bond here. So this bond, half of the bond is red, meaning it is oxygen, and this half of the bond is yellow, which is carbon. So carbon is generally gray, but here it is shown in yellow. So this is a carbon-oxygen bond. Then this bond is a carbon-nitrogen bond. Blue is for nitrogen. So bonds are represented like this. Qt can also display electron density. We won't go into that now, which is the result of structure determination experiments such as X-ray crystallography and EM reconstruction. So the electron density can appear as a mesh. Qt provides extensive features for model building and refinement. That means adjusting the model to fit the electron density, which we just saw here. We adjusted initially the phenylalanine was facing this side, but we adjusted the model. So now it is fitting with the electron density. And we can validate the model that whether the atomic model is agreeing with the electron density observed, and whether the amino acids are falling within the allowed Ramachandran region. So we can validate the model using Qt. Now, this was an example where this kind of visualization software was useful. So we have studied this before, that you have penicillin, and if beta-lactamase is added to penicillin, it breaks this lactam ring. So this is a four-membered lactam ring, and beta-lactamase is basically breaking this bond. So it is becoming COH and NH. So then the penicillin is deactivated. So this is the reaction that is happening. Now, PDB files and MTZ files are available for the beta-lactamase with the penicillin and without the penicillin. So there is a change in conformation. So beta-lactamase is a protein, and there is a change in conformation of the protein when it is isolated, and when it interacts with penicillin, it changes into something else. So by studying the PDB and MTZ files, we can identify this change of conformation, and that will help us in designing drugs. Similarly, you can see this is another reaction that is happening. So you have, so this reaction only is elaborated here. So you have the penicillin, which is a fused 5 and 4-membered ring. And in beta-lactamase, there is a serine which attacks this amide bond, and it breaks the 4-membered ring. So this is called acylation, because instead of COOH, the COO is attached to this serine molecule. And you can have deacylation, where some other amino acid like lysine comes here, and you can see the acyl group. So this will break. This bond will break with the serine. This bond is breaking, and it is becoming a CO minus group. We don't need to go into the chemistry of this. You can just remember that when this complex with the penicillin is forming, there is a particular orientation of the beta-lactamase protein, and when it is free, there is another orientation. And by studying these two orientations, we can figure out that how the protein is acting. So having this kind of model building software helps us understand the change in conformation that is happening, and we can use it to design different drugs. And these model buildings are a gift to the structural biology and computational biology community. So now we will look at two visualization softwares. I already showed you Qt. Now I will show PyMol. So PyMol, if I open, I'll open the same PDB in PyMol. So I'll go to File, Open. I'm opening this PDB 1MBN, which is the myoglobin PDB we downloaded. You can see it is a PyMol. You can observe the PDB. You can turn it around in different ways. You can increase or decrease the size. Now it is telling if we want to choose the color, basically for any color, there is a RGB value, which is the red, green, blue value. So here the color that is there is green. Suppose I want to change it to red, then I have to give this command. This command, I'll just copy it and paste it in this command bar. So you can see I've pasted this command and I do enter. And then I will put the second command, which will change the color into the RGB values that I have defined. So I'm putting this command. You can see the color has become red. So you can find RGB values for any color. You can just search it. So let me just, suppose I want a pink color. I will write pink RGB value. And you will get this kind of three values, 255, 192, 203. You can just copy it. But remember to use third brackets. So the command has to use this kind of square brackets. And you can change, you can see the color has changed to red here. Next, if we want to see the electrostatics, what we do here, I want to see the electrostatics of this protein. I will go to Wizard, Demo, Electrostatics. So what this is showing is that wherever blue color is there, the charge is plus 0.4, and wherever red color is there, the charge is minus 3.6. So depending on the different shades of color, the protein surface has different charges. So suppose I have a ligand which is positive. The positive ligand will bind to a negative region. So most probably it will bind somewhere here. Or if I have a negative ligand, you can see in this molecule, this dark blue region is not there. So uh, this protein will mostly bind positive ligands, because you can see there are regions of dark red color where the high negative charge is there. Minus 3.6 is very high negative charge. So high negative charge is there. So it can bind any positive ligand. But you can see the positive charge is very low. It is 0.4. So this is not a good protein for binding a negative ligand. So I'll just repeat. If you have a positively charged ligand, it will bind to the negatively charged regions of the protein, these dark red regions. But if you have a negative ligand, you can see the maximum positive charge of this protein surface is 0.4. So negative ligand will not bind very strongly with the protein. And if it binds at all, it will bind somewhere here, where you can see there is still some positive charge. So this is the electrostatics on the surface of the protein. So I'll just end this. Sorry, it has hanged. Just 1 second. I'll open PyMol again. So we have seen how to change color in PyMol and how to see the electrostatics. Now we will look at how the side chains can be visualized. So basically, to visualize side chain, you have to type this command in the search command bar. So I've copied it from here and I'm pasting it here. And then I have to select the residue and right click. So suppose I want to see this residue. I will right click, go to show and choose sticks. So you can see this is a valine residue. You can see the side chain has been shown. It is a isopropyl group, so it's a valine residue. So like this, you can see the side chain of this residue. Let me choose the side chain of another residue which is close to it, say here somewhere. I will right click, show sticks. So this is a threonine residue. So you can see two residues it is showing now side by side. Like that, you can see the side chain, you can display the side chain of any residue. Now I want to measure the distance between two residues. So I will go to wizard, then measurement. So wizard, measurement, then click on two atoms whose distance you want to measure. So suppose I want to measure the distance between this oxygen and this carbon. You will click it and you can see one value has appeared, 5.1. So it is 5.1 angstrom. Now suppose I select a residue, just 1 second. So like this, you can see the distance between these two atoms. So that is another feature. You can, any residue, you can display the side chain of the residue and you can find distance between two atoms. Now suppose I want to save this image. Suppose I want to save this image which is showing 5.1 angstrom distance between these two atoms. Then I will go to display background white. So any paper, wherever you see PDB images, it is in a white background. So you have to first make the background white, display background white. So you can see, and I will orient it in a nice orientation which I want for my diagram. So suppose I orient it like this. Then I have to type in this command, which will give a high resolution image. So it is ray, and then, so this command has to be typed to get a high resolution image. I will type it here. So this resolution is, and finally, we have to right click. So this check background is there, right? So you just right click to remove the check background. And we want to save this image. So we will go to File, File, Save Image As, and then go to PNG. So File, Export Image As, and go to PNG. So we will save the image as, let's suppose I save it as test. Save. So now I can see that this image has been saved. So this has been saved as a PNG file and you can paste it. If you are putting it in your paper or something, you can paste it from here. So that is how the image can be saved in PyMol. Other things that can be done in PyMol is, suppose you have an oligomer. So what I just showed was a protein with a single chain. An oligomer means that there are multiple chains. So that also can be viewed in PyMol. you can view the chain separately. You can change the color of the chain separately. All that can be done. And you can align different proteins also in PyMol. So this green protein is a different PDB, and the blue protein is a different PDB. You can align and superimpose the proteins to see where the two proteins are differing. And that can help you give an get an idea about, so these two proteins, suppose they differ slightly in function. So understanding the difference in structure of the two proteins by overlapping them, once you understand where the structure is differing, then you can predict that why the function is different. So that also can be done using PyMol. And in Coot, we saw WinCoot, we opened Coot or WinCoot, we opened one PDB file, and we saw that we visualized in the PDB file. So this was the same myoglobin protein which was opened in PyMol. Basically, this was called a ribbon representation, where the amino acid peptide chain is shown in a ribbon-like diagram. And this in Coot, what is it shown is the ball and stick diagram, where each of the bonds and atoms are shown separately. It is the same molecule. One function in WinCoot is that you can make mutants. So suppose you have an amino acid somewhere, you can mutate it to another amino acid. So I will show you how to do that now. So we have opened the PDB file in WinCoot, and we will go draw and go to atom. So this is the PDB file in WinCoot. I will go to draw and then go to atom. Then I will click on the drop down menu and choose the residue I want to mutate. So draw, go to atom. There is a drop down menu beside. So this is a monomeric molecule. So only one chain is there. I will click on this drop down menu and I will choose one residue to mutate. Let me choose leucine 9. I've chosen it and I will do apply. So now it is showing where the leucine is appearing. It is, you can see it is, let me expand the structure. The leucine is appearing here. Next, I will, the chosen residue is shown. Then on the menu on the right hand side, I will choose simple mutate. This symbol you can see, it is when you bring the cursor on the symbol, it says simple mutate. I will click this and the cursor has now become a plus sign. That is what this is saying. The pointer is becoming a plus sign. Then I will click on the residue I want to mutate and choose the amino acid I want to change it into. So I want to mutate this leucine. So I will click on this leucine. Just 1 second, simple mutate. I'll click on the alpha carbon of this leucine. And suppose I want to change the leucine to a, let's go with histidine. You can see the leucine has now changed into a histidine. So I have mutated that particular residue in the protein. So WinCoot can be used to mutate the residue like this. Then you can save the modified file as a different PDB. Now in PyMol, we can open both the PDBs, the native PDB and the mutated PDB, and we can overlap both the PDBs. So we have the mutated PDB formed by WinCoot and the native PDB. We can overlap them in PyMol, and we can create an image like this in PowerPoint. So you must have seen in papers where there are images like this, where you have two superimposed proteins. So this is the original protein where glutamic acid was there at position 39. This has been mutated to lysine at position 39. So this mutation was done in WinCoot, and then it was opened in PyMol, and this image was taken. This image was pasted in PowerPoint, and the mutated protein image was also pasted in PowerPoint. And then these dotted lines were drawn to explain that glutamine has been mutated to lysine. So altogether, 3 softwares were used to create this image. WinCut was used to do the mutation. PyMol was used to get this kind of nice ribbon representation. And these dotted lines and putting the images together was done in PowerPoint. So very often in papers, you will see images like this. So that is all the slides I have discussing the lectures. Now I will go through some of the question answers. Just 1 second, there is some problem in my network. Yeah, sorry, there was some problem in my network. I think I've reconnected now. So the question 1 is, which used for the amino acid sequence of protein? So as we have seen, this is the FASTA format. If you remember the FASTA format, it begins with this sign, and then you have the PDB code, the chain identity, the name of the protein, and from which species this protein has come from. Then you have the 1-letter code of all the amino acids in the protein sequence. And finally, it ends with a star. So this is the basic makeup of a FASTA file. And so the FASTA file shows the amino acid sequence of the protein. What about the other options? .mtz, we have seen that represents the electron density, which is derived from the X-ray crystallography. So the electron density map is stored in .mtz. .xvg is a special file. So GROMACS is a molecular dynamics software. And when you do molecular dynamics of any protein, the resulting graph is stored in a .xvg file from the GROMACS software. So, and .org, it is a simple text format file, which is used for, it is not related to any structural biology uses. So .FASTA is the one which tells us about the amino acid sequence, and .mtz tells us about the electron density map. .xvg tells us about the molecular dynamics of the molecule, and .org is not involved in structural biology in any way. So for this particular question, the answer is the FASTA file, that is option C. Then you have the question, the header line in a FASTA starts with, this we just saw, it starts with this sign, that is again option C. So that is kind of like you have to maintain that format, otherwise it is not saved as a FASTA file. So here it is the option C, and it ends with a star. So you can see it ends with a star. So these are the 2 requirements. The third question is asking, which of the following is not a protein structure visualization tool? So the answer to that is VLC. We know that PyMol we just saw, we can see the protein structure using it. Coot, we have also seen the protein structure using it. Rasmol I have not showed, but I have discussed in one of the initial slides that Rasmol is one of the first protein visualization softwares. So if I'll just go back to that slide, you can see this was the slide about Rasmol. So Rasmol was the first protein visualization software introduced in 1993, where this kind of a space filling model was used, and the depth of the model was also specified using light colors and dark colors to show the 3-dimensional structure. So this was the first time the problem of depth perception was solved. So Rasmol is also a three-dimensional visualization software. So you can see only VLC. VLC is basically the VLC media player, which does not have anything to do with protein structure visualization. So the answer of which of the following is not a protein structure visualization tool is option B. The next question is asking how many characters are present in the PDB ID. So here the answer is 4. So if you remember the PDB ID, we just saw, again, I'll go back to this. 1 MBN was the PDB ID here. So there are 4 characters. That is what the question is asking. How many characters are there in a PDB ID? There are 4. So the answer is B, that is 4. The next question is asking, in PDB file, there was a term called connect. And what does it define? What does this term connect define? So what it defines is all of the above. After connect, if there are 2 terms, it defines a bonded interaction. So atom number 1853 and 1852 have a bond between them. After the term connect, if there are 3 numbers, it defines an angle. So atom 1859, 1858 and 1860 have an angle between them. And if there are 4 numbers after the term connect, it defines a dihedral angle. So atoms 1856, 1851, 1854 and 1857 are connected to form a dihedral angle. So the term connect defines bonded interaction, it defines angles, and it defines dihedral. So it defines all of the above. So the correct answer is D. The next question is asking, which command or tool is used for high definition images in PyMol? So here the answer is Ray. So I just showed you in PyMol. I'll go to PyMol again. If you remember, we had given this command, Ray 1080720, to generate a high resolution image. And after that image was rendered, we saved the image as a PNG. So the command used to define a high definition image is RAY or Ray. That is option B. The next question is asking, which of the following is present in the model fit or refinement tool of COOT? So here the answer is simple mutate. So if you remember what we did here, we showed how mutation of an amino acid can be done using COOT. So this was initially a leucine and we have mutated it to histidine. That is what I showed. So using this kind of mutation, you can, one second. So if you have an electron density map, suppose you don't know what amino acid will fit into it, you can mutate the amino acid into different residues and see which amino acid has the closest fit to the electron density map obtained from X-ray crystallography. So using the simple mutate tool, you can fit the protein structure with the electron density map. So that is what the question is asking. For model, fit, or refinement, which function can you use? It is the simple mutate function. Because using the simple mutate function, you can change one amino acid into another, and you can fit the model to the observed electron density map. So the answer is B. The next question is asking, the measurement displayed between two atoms in pi mole has what unit? So here the answer is angstrom. So again, I'll go back to pi mole. If you see this here, 5.1, 5.1 distance between this carbon and this oxygen. So what is the unit of this? It is in atomic units, which is, generally, uh, the default is angstrom. So this 5.1 refers to angstrom. So the answer is C, angstrom. Then the question is asking, which of the following parameters one cannot get from validation report from the Worldwide Protein Data Bank? So the answer is macromolecule fold. Let me go back to the validation report slide. You can see in the validation report, nowhere the fold is mentioned. You have R value, Ramachandran outliers, different things. You have the amino acid sequence and the matches with the X-ray crystal structure. But nowhere here do you have the macromolecule fold. So now I will again go back to the question. So entry, composition, data, refine statistics, resolution, model quality. Entry composition, it tells you the amino acid sequence. Refine characteristics, clearly all these things are telling how refined your structure is, and also the validity of the structure. And resolution and model quality. Resolution and model quality you can understand from this report. What is the, so you can, as I said, the left one was a good model, right one was not a good model. And resolution, you can see it is given 2 angstrom. So in the PDB itself, you will have that information. But in this report, nowhere the macromolecule fold is mentioned. So that is the information that is not gotten, which is the macromolecule fold, that is D. Next question is asking, what is the format for the command of RGB color in PyMol? So the format I discussed previously also is this third bracket, this square bracket, that is option C. So if I go back to that slide, you can see we have to give 2 commands. So I'll just reduce this. Let me give the commands here. You can see the color changed. So what did we use? We use square brackets to give the RGB values. So the answer is square brackets have to be used. The next question is that which is the important software to make diagrams? So the answer is all of the above. So if we go back to make this kind of a diagram, initially Wincott had been used to mutate the amino acid from glutamine to lysine. Then the modified PDB had been saved. Then the native PDB had been opened in PyMol, and we saw how to save pictures in PyMol. So we can save pictures in PyMol. I showed how. So we take the picture of the initial PDB and the mutated PDB in PyMol, and we paste it in a PowerPoint presentation, and then we can highlight using dotted lines like this. And this kind of image you will see appears in paper many times. It shows which region the mutation has been done. So to create image, you used Wincott for the mutation, PyMol to get the image, and PowerPoint presentation to put the image together. So all three software were used. That is what this question is asking, that which software is used to make the diagrams? The answer is all of the above. The next question is saying, which experimental structure cannot be deposited in PDB? So here the answer is comparative modeling. So we know that X-ray crystallography has been used many times, and you can see also this structure, X-ray diffraction, has been used. Now, if we go to more recent structures, you will see NMR has also been used. Now, mass spectroscopy on its own cannot be used, but in the structures deposited in PDB, the mass information is provided. So mass spectrometry is you can think of it that like X-ray crystallography is a standalone technique. Just using it, you can get the structure, or just using NMR, you can get the structure. So A and B are standalone techniques. That is, on its own, it is sufficient to give at least a basic structure before refinement. Mass spectrometry cannot do that, but it is one additional information that is included in the PDB. So this experimental information is included in the PDB. But comparative modeling, so comparing two PDBs, you cannot deposit that in the protein data bank. Protein data bank each has to be a unique three-dimensional structure of a protein. You cannot compare two proteins and then deposit it in PDB separately. So number C is not available in PDB. So the correct answer is option C. Then the question is asking, which of the following is incorrect about Swiss PDB Viewer? That is another visualization software which is commonly used. So here the answer is B. So basically, Swiss PDB Viewer is a multi-platform viewer. That means it can be used with different, this is true. This means that it can be used in different operating systems like Windows, Mac, Linux, and so on. So if one is true, B has to be false, because if it is multi-platform, it's definitely not single platform. Option C is saying it is capable of structure visualization, analysis, and homology modeling. This is also true. Swiss PDB Viewer is like PyMol. You can observe the structure, you can analyze the different amino acids distances, you can superimpose two structures. So C is also true. So which of the following is incorrect is your option B. And the last question for today is that what is the general representation of PDB where X is an integer and B, C, D are any alphabet from A to Z? Just imagine that it can be any alphabet. So the general representation is A, where the first character is a number, an integer, which is the X, and the alphabets B, C, D come later. So you can see that also. First is an integer, and the next three are alphabets, M, B, N. Let me search for another PDB. Let's suppose hemoglobin. I can just type the name here, hemoglobin. So you can see that numbers, so numbers can come in the fourth position also. So is that there in any of the options? It is not there. Number can come in the, so only the second position cannot, so first position has to be a number. The rest of the three, they can be alphabets. The only two rules is that the first position has to be a number and the second position has to be an alphabet. So let us look at the options. First, so first position has to be a number. That means automatically B and D get cancelled out because first position is not a number. The second rule is second position has to be an alphabet. So C also gets cancelled out. A is your correct answer. Another possibility is X, B, X, T. This is also a valid, but it is not there in the options. Or it can be X, B, C, X. This is also a valid code, but it is again not there in the options. You can see, you can have a number in the fourth position, number in third position. So the only rule to remember is that first position has to be a number, second position has to be an alphabet. These are the two rules to remember when a PDB ID is being defined. So, and any PDB, even if you don't know the PDB, you can just search the name of the protein here and you will get the PDB. Then you can click on the structure and you will be able to, this is the hemoglobin of this particular organism. And you can go to download files and download the PDB format. And the PDB format can be opened in PyMol, Qt, and it can be visualized. So that is all about the different visualization softwares, which was covered in week 9. And as always, I will be uploading both the slides and the PPT, so you can check it out and come back with your doubts later. Thank you for attending the class. I'll end it here.